The Centre presents a new podcast, CBME and You, a podcast devoted to promoting awareness and debate around controversial ethical issues in biomedicine.
And welcome back to CBME and You, a podcast about biomedical ethics. Today, we dive into the strange, wonderful world of organoids. What are they? Why would we want to make them? And do we get to talk about an all-time philosophical hit, Brains and Vats? I'm excited. Joining us for today's pod is Professor Insoo Hyun, senior researcher at the Hastings Center for Bioethics and affiliate of the Center for Bioethics at the Harvard Medical School. Bam, first try.
You
did it. It's a mouthful.
Welcome.
Thank you.
We have a couple of questions to
Sure.
get to know you a little bit better. First question, I see your hand covering the cup of coffee. Coffee or copy?
That's a really good question. So I would say, thank you, first of all, for letting me be here. This is really fun. But second thing I want to say, the very important question. So health-wise, I would prefer coffee. But if you actually put down in front of me a cup of coffee and a cup of coffee, I would grab the coffee.
Really?
So I think if your preferences are revealed by behavior, that reveals what I prefer.
I'm so intrigued because this is the first time you've had copy. And so you're saying your preferences have changed that quickly. Are you trying to suck up to us?
Well, no. I think coffee is pretty similar to other things I like Vietnamese coffee. And there are many coffees that have that sweet condensed milk added to it. I don't think I could drink it daily. So like I said, for health reasons, I think coffee is a better choice. But again, if you put 2 cups in front of me, the hand will grab the cuppy.
The hand. Not you, the hand.
The hand, yeah. It just inexplicably grabs
He's like a behaviorist about himself.
Exactly, yeah. You can talk big about what your values are, but really it's what you do that I think
Talk is cheap.
reveals your true preferences.
I agree. I think coffee is delicious. When I first moved here, I had one every day until A, I realized how much sugar is actually in it, and B, how much caffeine is truly in it. And now I sort of treat it like a sweet treat. When I need just a little bit of caffeine, but I want something sweet, coffee is where I go. But you definitely have to have it before midday, otherwise I will be up all night.
Yeah, I think I've passed the midday point, so thank you for this. I'll be lying in bed tonight just cursing the two of you.
You wouldn't be the first.
Your curses will be well received.
Okay.
Next question. What country do you want to travel to next?
Yeah, it's actually the country I'm going to next. Next week, I'm going to Kenya.
Wow.
And it'll be my first time, and I'm really looking forward to it. I'll spend some extra time on safari, not shooting anything, but, obviously just watching, looking for animals.
I'm sorry, not in the gun context. I thought you meant shooting a movie.
No. That would be kind of fun too, but no. Just iPhone capture of animals.
Yeah.
So I'm really looking forward to that. I've always wanted to go, and I have an opportunity to go for work reasons. But again, you add on days to make it fun. Yeah.
Well, I hope the iPhone shooting goes really well. We look forward to seeing your post online.
I hope you
all these blurry shots of these little specks. How
Zoomed
wonderful!
in.
Yeah.
How wonderful is this picture? It's what is that? A rat?
If you squint, you might be able to see the lion.
Right.
The lion that was 2 kilometers
Move the picture
away.
in front of your face really fast, because that's what it was like in real life.
Well, there's a effect now on Instagram that's really popular that actually blurs it to the kind of quality of photos that were taken in the 2000s. So we're so nostalgic for better times that we are actively making our
Downgrade
photos
the
uglier, worse quality.
There was a time where you actually had to take photos and hope that it turned out, and you have to wait for the developer to develop them. It does kind of make each shot sort of precious, right? You have to be really thoughtful. It's not just volume, and then you pick the one that looks good out of 100, but you could end up getting 12 back, and they're all terrible. That's the risk.
That is quite sad.
Yeah.
Last question for the whole entire pod, and then you can go home. What's your go-to karaoke song?
So I'm a terrible singer. It's one of the things I wish I could sing. Probably, but anyway. So I'm a terrible singer. But if I had to do karaoke with a gun to my head, I probably pick a song that's I think it'd be pretty easy for me. It's Billy Idol's "Eyes Without a Face" because it's kind of monotone. It's not there's not a lot of range to that song. It's kind of
Can you give us,
mid-level.
a little demonstration?
No, I can't because you don't have a gun to my head. There's no incentive for me to embarrass
You
myself.
give the
We have cameras.
qualifier.
We could shoot you.
That's true.
with the
Right?
cameras.
I'll be real blurry.
Yeah.
But that would be it. My wife is a fantastic singer, and her go-to is either. I actually recently it's been that song by Four Non Blondes, "What's Up."
Oh, and
Yeah,
I
she
say, hey.
That one, yeah.
Oh,
She and her female friends just get all into it. That's fun. So I watch that, but I don't embarrass myself. Try not to.
Just so you know, out of the two of us,
Mm-hmm.
James is the better karaoke person at all types of singing, including
Really?
higher pitches, which is very disappointing for me because it's like, what do I contribute to karaoke if he can do all the high
Yeah.
bits?
Well, here at the center, we believe that karaoke is not about singing ability. It's about the
Vibes.
Interesting. Okay.
Now, I can't say I'll put a gun to your head one day, but I do hope that when you come back to Singapore, we can entice you to do karaoke.
I'll do the Billy Idol song. Go back to 1984 or 83, I think is when it came out.
Okay, so in the words of one of my favorite karaoke songs, let's get down to business.
Oh,
I like what you did there.
I like it too. Leave it in. Okay, so let's start with the basic question. Many people are wondering right now, what are these organoids that we're about to talk about?
Yeah, so there are various ways to explain it. So organoids are many, many different types. Basically, there are three-dimensional models of human organ systems. So there are brain organoids, there are liver organoids, airway, eye, gut, you name it. And they're small. Right? So, I mean, the idea of calling something an organoid it may make people envision kind of like a really sizable clump of tissue. But for example, human brain organoids are really no bigger than boba, and they kind of look like white boba. I wouldn't recommend putting that in your tea, but that's what they look like. But when you imagine, maybe you hear the term brain organoid, you kind of think, oh, it might be pink and has like folds and it looks like a little mini tiny brain. Doesn't look like that at all. Now, how are they made? You can make them from what are called pluripotent stem cells, which are stem cells that could become any kind of tissue type, any cell type. And traditionally, we've started by using embryonic stem cells, which come from fertility clinic embryos, pre-implantation. Now we can make those types of cells from any kind of cell from an adult body. So we can use blood cells, skin cells. You reprogram them in the lab to become pluripotent. And then once you get a pluripotent cell line from a sample, you can coax it, you can nudge it to make these different organoids. And it's really quite amazing. I mean, you can make these models in a dish, and they are pretty consistent in terms of how they, to use a fancy term, recapitulate developmental biology or replay the developmental playbook of like when your brain forms or when other organ systems form. So it's really a cool tool to see how someone with a genetic disorder might be different in terms of their early development by looking at these different organ systems compared to a control cell line, a quote-unquote healthy cell line and what it does. So it's a really beautiful model, three-dimensional of organ systems to study function, growth, and also compare diseased and non-diseased organoids. And then the application for this, besides expanding your knowledge of what cells do and what development can do, is to actually have a three-dimensional test bed for drugs and other interventions. So if you make a disease-specific, let's say brain organoid that comes from an autism patient, you can try to see like what's not only different about the neuronal growth, but are there any factors you could apply to the brain organoid to slow down or reverse the abnormalities there. So as a research tool, very, very interesting. They don't look like anything impressive. Like I said, the brain organoids kind of look like boba floating
I'm a
around.
little bit disappointed about that.
A little disappointed, yeah. But when you put it under a microscope after you've tagged all the cell types with different colors, it's really beautiful. So anybody at home, Google brain organoid, and what you'll see in the images are just beautiful rainbow-colored things that under a black light will sort of shine and show all the different cell types. But normally when you look at them in a dish or a vial, They just look like little white blobs.
And so these are literally living things. Like
That's a good point. Yes, it's living cells, and they actually go through the initial stages of embryo and fetal development. So brain organoids could be grown or kept in a dish, growing, fed for a year or longer. Now, it might take 9 months to a year to start seeing mature brain cell types because the actual developmental program in humans is shown yet again in the dish. But now, why are they so small? Why are they white? It's because they don't have vasculature. And because there's no way to bring in nutrients and remove waste products, that really limits how big they can get. Because once they start growing beyond a certain size, the very middle core of the brain organoid starts to die. It forms what's called a necrotic core. And so it really is kind of limited in terms of how large they can get. There are people who are trying to find ways to vascularize them, or this is really fascinating, combine them with other organoid types. They could be the same brain organoid regions, or it can be other organ types. So that combination is what we call assembloids. You take brain organoids and maybe other brain organoids, and you put them together, and it forms a little bit more of a complex fused model. The thing I should point out is brain organoids only recapitulate a particular region of the brain. So you never have a single brain organoid that represents everything. It's going to be like, cerebellum or frontal cortex or some other part. And so the assembloid idea is kind of cool because you can imagine it's almost like taking little Play-Doh balls and you put them together and kind of assemble this little bit of a approximation of the real brain. So they get a little bit bigger. The really fascinating work is where you combine organoids of different types. So one research team that I've worked with a lot at Stanford, Sergio Pashka's team, he put a brain organoid, fused it with a spinal cord organoid, and then put on the end of that a muscle organoid, and it got the muscle organoid to twitch by stimulating the brain organoid. Now, why is that important? Well, there could be, a really interesting model system for movement disorders or other things, and, you can get these cells from people who have various conditions you want to study, and then you just basically build a little model of their physiology. It's so interesting because it's genetically specific to that person. So you can make it really disease-specific, individual specific. So that's the kind of work. There are other, we can talk later about the other, what are the issues coming up on that? Because that's been a big area of my research. What are the other issues, ethical issues, whether it's about, worries about consciousness or other applications of them. So happy to get into that later. But a very long response or answer to your question, what are organoids? There are these things that are created in the lab, three-dimensional, used for all these different types of purposes.
I can just imagine my brain is going, a lab scientist has two organoids, and it's like, now kiss, and just puts them together.
Yeah, sometimes I ask people, and they don't give me a straight answer. I say, do you have your entire lab? Did everybody in your lab contribute materials, and you have organoids, and you have a culture dish with your whole lab represented in organoids? I think probably somewhere people have done that because, lab folks are kind of fun and quirky. and I'm sure people will be, because it's easy to do. You can just take a blood draw, you can take a little skin sample, you reprogram that into what are called induced pluripotent stem cells, iPS cells. You can do it yourself. I wouldn't really call it self-experimentation, but it's just sort of, you're at the lab, you're doing this all day with other samples, why not just throw in your own sample and do it? I wouldn't tell on them if that's what they did, but I wouldn't be surprised. So the next step is then what do you do with that cell line? Maybe you enter it into the protocol that you're working on and you just see, what do my brain organoids look like? Wouldn't you be curious?
Yeah.
And look at it under the microscope and say, this is replaying what happened in the womb when I was coming into being.
Yeah. I'd be very scared though. What if my brain organoid turns out to be like, bleh.
Well, what's interesting? It's they have found some strange, and you don't know why, but some cell lines don't do a good job making organoids. It's why? Is it because there's something intrinsic about the cell line that really, it hints at a potential problem that you may be facing? One of the interesting ethical issues in this area of work, so I've worked with these labs. We had a grant from the NIH to look at ethical issues and scientific issues early on in brain organoid research. And one of the things we've realized was there could be what we might call developmental incidental findings. So you guys know what incidental findings is in terms of genetics research and imaging. You during the course of your research might have something like, CT scans. You could have genetic samples, and you're, probing around, getting your data, and then nearby, you realize there's some abnormality. And you wonder, in this scan, it looks like they might have like a tumor. Or in this, genetic panel that we're doing, they actually have a mutation for a pretty serious disease. I don't know who this person is. I don't know anything about this person, but I'm working with their biospecimen or their image. Do I have a responsibility to return this information back to them and give them a heads up? Hey, you may want to go to the doctor and get a checkup. So that's a whole issue of incidental findings in genetics and imaging. What's interesting about the brain organoid stuff or other brain organoid research is there might be some issues that you're seeing in the developmental trajectory that might hint at something unusual about the person's brain or about the person's gut or anything like that. And do you have a responsibility to go back to the original donor and not you yourself, but maybe contact the clinician in charge who really is supposed to be the person who talks to the original donor? And the answers for all these issues normally is, well, you better make sure that you validate what you found clinically. You can't just kind of take a research grade scan and scare everybody, and waste time and money following up if it's not really like it's not clinical level. And so the problem is, well, how do we know that the brain organoid is truly, truly representative of development? We're still figuring that out. So it's too early to do anything about that. But it is really interesting, You say, yeah, what if you find something strange? And people have found unusual differences between the organoid types. And again, you don't know exactly what that signifies. But maybe in the future, once we know that these models are really great models, you may be in a position where, that does become a serious consideration. You need to get somebody needs to get in touch with this person, especially if it's actionable, like they can do something about it. There's another area where people might argue if you can't do anything about it, why would you want to tell them? And there are actually some donors who say, I wouldn't want to know if it was something you found that I couldn't do anything about.
James, would you want to know when they find something?
I think I would. Unless it's something really weird, then, don't tell me. Not like a health issue, but like, turns out like, oh yeah, you should have, you have scales or something. You would be growing scales.
Yeah, here's the problem with that. So you tell the researcher, well, don't tell me anything if it's really bad or weird. And the person says, oh, okay, I won't tell you anything then.
It's like, well, guess what? You're all weird. Everything is. This is really interesting because I'm now wondering what the limits are to this, right?
Mm-hmm.
Like, so you've already mentioned, well, it's going to be really hard to advance in terms of size, at least, if we don't find a way to vascularize these organoids. But now I'm wondering, both in terms of practical limits and theoretical limits, looking into the future. Let's do some sci-fi projection here. What are the limits here? Could we actually just like grow full brains or parts of brains and like have them hovering around in vats?
Yeah, I think there are lots of technical limitations happening here. So we still haven't solved the vascularization problem, which is a big hindrance in how big and complex these things can get. The other one is people worry. So let's just stick with brain organoids because of all the organoid types, that seems to be the one that's the most controversial. I think people are more concerned about brain organoids because brains represent personhood and seats of consciousness. If you're modeling that, then it seems like the concerns and all the value we place on human brains gets transferred over into these models. Whereas the gut, sort of, who cares about the gut or the kidney morally? I mean, except for like maybe transplant reasons. Let me qualify that. There are two other areas. So I think it's brain organoids and what are called gonadal organoids, or organoids of testes and the ovaries. Seems to be like brains and gonads are all what people care about or worry about. So there are people who are trying to develop testicular organoids and ovarian ones, and I can say like why. I mean, why would they do that in a minute? But let's just stick with the brain organoids. So a couple of worries about the future. One is right now, what we found in our brain organoid project for the NIH was we don't have to worry right now about there being consciousness or sentience in these little boba balls in a vat. These are like basically brain models in a vat, right? They're floating around solution. Why? Well, we can give a number of answers to say why right now we're not worried about anything like consciousness. There are just 1 million or 2 million cells in a brain organoid. The human brain has You can look it up. I don't remember the exact number, but it's billions of cells. Not only in volume, it's you don't have anything close to the real human brain, but it doesn't have the regions you need, all the different regions. It doesn't have long range projection. Everything that we know about consciousness today has to do with kind of the architecture of the brain, not just that there are neurons, but it's the architecture, it's the arrangement, it's the long-term signaling. The brain starts to develop in combination with interaction with the environment, right? So you can imagine, this is a terrible thing to imagine, but you can imagine like a newborn baby being born and just like kept in a vat. I don't think that baby could develop what we would consider to be typical or normal human awareness and consciousness.
Baby has environmental.
So that's what's happening during pregnancy. You got a little, brain in a vat with everything else, right? But it still has limbs. It has other nerves, and there's some interaction with the maternal environment, obviously, right? And babies can. Or fetuses in utero can hear.
Yeah, they can hear.
There are lots of things, right? So there are no sensory organs. There's no input like that. So it's just floating around. But getting back to the newborn idea, even with all the other sensory inputs and stuff, just without having social interaction, without having any kind of communication, you miss a window of opportunity in developmental brain biology to actually develop the capacity for language and for recognition and empathy and all the things that make us human. So just, you can't recreate those circumstances in the brain organ in the dish. Now, I say that, for right now, we don't really, worry about consciousness, but there are people, and I'm on one of these projects where they want to do brain organoid-based computing. So you use a brain organoid instead of a microchip to do some basic computation for, let's say, AI applications. And, so there's some interaction with the outside world, this little neural network kind of, getting some stimulation and having output. Still, you think, well, that's still kind of like brain into that kind of work. So what the team has been thinking of doing is maybe pairing the brain organoid with a humanoid robot, and it communicates through Bluetooth. And so then you kind of give it's not physically inside the robot, but it's interacting with a body, it's got embodied. And that's kind of a couple of things, just to kind of see, can you develop neurons more accurately through this kind of interaction with an environment. And then, can the brain organoid kind of like it does in more like quote unquote desktop computing, like control something like that? And they're finding that it can.
How does a brain organoid, like a boba, connect via Bluetooth to a robot?
Yeah. So it sits on top of, it's called wetware instead of hardware. And so you can look up papers. So if you Google, telling people to Google lots of stuff. If you Google organoid wetware or organoid-based computing, the images will show from the scientific papers. There was a proof of principle paper published in Nature Electronics, and that team is the team I'm working with. So in their figures, it should be available online. You'll see what these things look like. They're drawings, schematics, but I actually honestly don't quite understand how it works, but it seems to work.
James, you could finally have a friend.
It could even be your little organoid.
The funny thing is, like,
it could just be you.
I was just thinking of like a video game which I think uses the term wetware. I think it's like Cyberpunk or something. I was like, well, so guess what? Video games are already my friend.
Really happy for you.
Yeah.
People obviously donate cells to create lots of these research lines and cell lines ethically, hopefully now. It's not necessarily just lab scientists using their own cells for all of this. So are you finding that people are more concerned about donating cells when it could be growing into something alive?
Yeah. So there are two ways to answer that. So one is, unfortunately, a lot of the work that's being done in this area is with pretty long established cell lines for which there was no explicit consent for this kind of work.
M-cell?
They've used, there are so many different ones. So yeah, I wouldn't be surprised if that's one of them. But so what that means is there is no explicit consent for anything like this kind of work. And the reason why people can sort of get away with this is you've anonymized the cell line, meaning you scrubbed all the personal identifying information off that. My view is, genetics is the ultimate personal identifier. So actually, if you take any of these cell lines, if you really wanted to, you could probably figure out who that is, right? So I think that idea of that little quote unquote loophole of not having to get explicit consent or even any consent from people who provided tissues during hospital stays and other things like that is probably on the way out. It's going to be harder and harder to defend this idea of, well, you scrubbed away their personal information, there's no risk of privacy invasion, I think, because all these cell lines, all the organoids have also genetic data attached to them, and it travels with the cell line. It just takes someone just, the interest and the motivation just to triage, from the sequencing all the different like publicly available or commercially available ancestry databases, and you just figure it out. And people have been able to do that. So one way to answer your question is, so far there actually hasn't been a very robust history of getting consent for this kind of work. I mentioned the hospitals. This is something that I should probably point out to your listeners, and they might be interested in this. Anytime you go get medical care at a teaching hospital, so it's a hospital like at NUS or hospital Harvard Medical School, whatever, that's affiliated with a research institution or a medical school, what you'll notice is that there's a consent form for when you get admitted to the hospital. And so you're consenting to, being treated at the hospital, et cetera. There's usually a paragraph in there that says anything I leave behind, meaning like any biomaterials you leave behind in the hospital, whether it's urine in a urine bag, and yes, you can get iPS cells from urine because there are cells in your urine, or, a blood draw, any clinical discards of tissue during your procedure, anything you leave behind can be used for education and research. That's what it says in the consent form. It can be used for education and research at that institution.
Does that include if you just go to the toilet?
So fecal matter is not great because it's hard to isolate DNA because there's so much microbial things in it, so yuck, right? But the urine is sterile, and there are a lot of cells floating around in the urine from your bladder. So that's actually probably the least invasive way to get cells for your research. Yeah, so hold your pee when you're at the doctor.
Make it to the MRT.
You insist that you're going to flush it down the toilet. You're not going to leave it in the bag behind because they're going to take that little bag and make iPS cells and brain organoids from it.
So not to raise any panic, but for all we know, there could be like mini Jimmys that somebody is like growing somewhere.
So that's interesting. Yeah, that's interesting. And the pee. Yes, I mean it could in principle be the case, and you would think, if I found that out, would I be satisfied with the rationale that, well, don't worry, we scrubbed your identifying information off that? You might think, well, no, but because the way you said it, could there be something that I'm kind of like tied to biologically, right?
Yeah.
You said mini U's, but so I think that's of great interest to me. I've asked the lawyer, so what is someone supposed to do to opt out of this? And I've been in the medical field. I've been doing research ethics for a long time. The answer really surprised me. She said, you would have to find that paragraph, circle it, cross it out, write no, and initial it. Who the heck would know how to do that? I didn't know.
Yeah.
Right? So I had argued, you need to actually have institutional policy that you will not use those specimens for this type of work because it's so far beyond what people imagine could happen. Can you imagine telling somebody, some patient who's at the hospital getting their routine care or getting their surgery, and you say, you point out, anything you leave behind here could be used for education and research. Oh, yeah, that's fine. Helps people. That's cool. Why would I care? What you do with my
cure cancer,
discard.
Please.
Yeah, and then he said, do you know that one of the things you could do for education and research, and this might be a very active area in this particular institution, is to convert some of these cells into an immortal cell line. What's that? Well, this is a perpetual stem cell line, can be shared around the world, has all your genetic data, and people can do other things with it. Of course, it's all reviewed by a committee, but the future is pretty wide open for that. Oh, well, what are some of the crazy things that could happen? Ever heard of brain organoids? So can you imagine somebody sitting there contemplating that that's one possibility? Now, I want to tell the audience, it's not like it's super common, but there are some places for which there's a lot of this research happening, so chances are not zero that this could happen. So I think that that's a big problem. When you get back to of course you would have to get or want to get consent explicitly for this kind of work. When you talk to those people, like the prospective donors, they do say, I would want to know if what you're going to do, one of the things you're going to do is brain organoids. And you say, well, why? Because I feel this sort of connection maybe to this, and it's important that I'm at least aware of what you're doing, why you're doing it, what's going to happen to the brain organoids. It's a little interesting to me because it's sort of like you can't have your cake and eat it too as a researcher. You can't say, on the one hand, this is such an incredible model because it tells me what happened when you were developing. Because that's what they tell like autism, patients, they tell like parents who are donating samples. They'll say one of the goals of this research is to better understand something about your child. So you want to, that's true. But you can't at the same time then say to the other donors, oh, don't worry, it's just a lump of tissue. It's meaningless. How could you have it both ways? Either where it's super significant and biologically really it is tied to their history in some way, their developmental history, or it's just a mere lump of worthless tissue. You can't say both. So that I see the attitude kind of changing. Like on the one hand, if you're talking to some donors, you really want to like convey the truth, which is like this is pretty significant in terms of what it will tell us about development. And then if you want to kind of say, oh, no, there's nothing to worry about, then you end up downplaying what's exactly so scientifically compelling about this work. So anyway, so I think that the folks who do say I would want to know, they do have grounds for kind of, having an interest in what happens to them, their cell lines. And we've done research that we've published papers on like donors' attitudes toward organoid research, and especially for brain organoids, they want to know. So it kind of backs up what I said earlier about, sometimes people don't care so much about other things that don't have this like moral or sentimental connection, whether it's the gut or liver. I mean, it's interesting. Some people have some connection to eye cup, or heart. It's really kind of interesting culturally, right? You kind of say, go through the organ system, which ones kind of matter to you?
My colon.
Yeah.
I'm just kidding.
Yeah. Well, there's some people for whom that's
I believed you.
That's a big deal.
I was so laughing. You guys were like, Can I ask for both of you? Do you feel strange about having a brain organoid based on your own DNA?
Would I? I don't think I have any in the world, but would I wouldn't feel strange. I would be really fascinated. Like I really want to look at it.
Just look at it.
And yeah, I mean, just
Look it a little.
Yeah, I'd be interested.
Yeah,
I would.
Yeah, I'd be fascinated. I'd also be a bit flattered, if
a bit flattered.
like if a bunch of team of scientists told me that they wanted to take my cells and like make it into the immortal gene
what
line,
about put it in the robot?
said.
You want to do the robot stuff?
Yeah, and then like send it all over the world and be like, me?
you just like hold your head a little bit high when you walk.
You're
around.
going big.
Yeah, well,
Mr. Worldwide.
Okay.
So is there research asking people how they feel about this, or is it just a presupposition that because it's the brain?
I understand the question. So it was a study where we asked people
how they feel about it.
Yes.
And the majority of people feel like there's a special something about it.
Yeah. People may have heard of the Gila cell line, the Henrietta Lacks cell line. And it's fascinating because her family still talks about that cell line as if it had some meaningful connection to their loved one. They'll say, our grandmother has been to space. Our grandmother has been in labs all over the world. She has helped, develop a vaccine for polio, which is all true. But they're saying she was part of that, and that's even that's not even a neural cell. It's a lump of cervical cancers, tumor cells that are just growing like crazy. So normally people don't identify with your tumor cells, right? Probably more so with like heart cells and, like I said, the brain. That way of thinking outside of the scientific community is prevalent. People do think that there's this connection to it. When you talk to donor families for let's say the autism studies, a lot of them want to know so you just told us that this is really valuable. It's kind of unlocking a lot of information about our child who in a lot of cases, these are for undiagnosable neurological disorders. Are just trying to figure out what is going on here. So you just told us about all the relevant biological connections to them. What are you going to do with them after you're done? How are you going to dispose of them? It's really kind of an interesting question, right? Are you going to treat it respectfully? It's not. It shows me that there's again great importance that maybe scientists overlook us. They're just so familiar with this stuff. It's kind of routine. For the person just hearing about this for the first time, it kind of framing it in their own ways of understanding the world and their loved ones. It's quite remarkable that other people may not think that that's something that you have to talk about, but clearly from the donors that they have these questions that the IRB or other groups wouldn't have necessarily thought should be a talking point. Yeah,
it kind of sounds like is there a TV series called Black Friday or like the Netflix horror series.
Black Mirror?
Black Mirror. Thank you. Friday, the scariest thing I could think of.
Big content consumer, this one.
Massive. I feel like it could be an episode of Black Mirror where I think there was an episode where you could order a replication of a loved one who'd passed away to try to help you deal with the grieving process. I wonder if we would get to a point where you could get like a little organoid of a loved one, and you just like feed it, or we could teach people how to, because you feed your cells.
Well,
You hydrate them.
I'll do one better than that, because one of the things I didn't talk about, because you asked me what are organoids, and I told you about organoids, I told you about assembloids, which are pretty crude. There's actually embryo models you can make with stem cells. So what you do is it's not just doesn't just make a brain organoid or a heart organ. It does the whole model, the whole embryo, both pre-implantation stage and we think post-implantation stage.
But we haven't tested it in a womb.
No, and so the guidelines that I helped draft internationally say you can't transfer any human embryo models into a human or non-human uterus. But I think we have to revisit that language, because now there are teams that will make these embryo models. So again, the idea of like taking cells from you, programming them into iPS cells, you put them together and they self-organize. All these things are just incredible. They self-organize. They know what to do into what look like pre-implantation stage fertility clinic embryos. Those are what are called blastocysts. These are what they call blastoids. They look a lot like that. Are they able to make a baby? Well, you make mouse blastoids from mouse cells, transplant them in a uterus, and it kind of starts triggering a pregnancy response, but they don't go very far. And there's some scientific reasons I'm not going to get into, like technical reasons for why they think developmentally they don't go so far. It's about erasing parental imprinting. But anyway, it's incredible work. So you were saying, what if someone were to kind of like someone passed away, and you've in principle, I'm not saying that this is what anybody should do. But in principle, the technology is there where your loved one has passed away. You take a little bit of skin, any biospecimen of living cells, send it to the lab. They make iPS cells, and then they can make not one, but countless, as many as you want, embryo models that are genetically identical to that person. Now, this to me brings up the idea of human cloning, where originally the idea of cloning was you take an unfertilized egg, you take out the nucleus, and you put it in the nucleus of a somatic cell. That's how they clone dolly to sheep. It's how they clone cats and other animals today. But the thing about that is you have different mitochondria, because you have the mitochondria from the donor egg. So it's not a true clone of the original. And in fact, having different mitochondria might affect development in interesting ways. This case is everything's the same. It's all the same mitochondria. It's everything. It's
Like make a Sinead 2.0.
Yeah. So you can create embryo models and
you can have a friend. You can
store
make
them.
your own friend. Oh wait, you're not a woman.
And you can store them. Yeah. Yeah. Here's another wild one. So other things that you can do in stem cell biology. This is the thing I find fascinating about stem cell biology is that it kind of makes human cells and human biomaterial, quote unquote, plastic, which means you can mold it into other things. You can do things with it. You can build things with it. Okay? Because you transfer the cells back to an original cell type, a blank slate called a pluripotent stem cell, and then you drive it down, make it do other things. So you can build a lot of things with that same DNA.
You can replace your own heart.
Yeah, I mean, one of the dreams of this research is to have personalized regenerative medicine, right? But one of the things that's really interesting is not only can you make stem cells, make organoids and brain cells and all that, but you can also, in principle, create functional gametes, sperm or eggs. So they've done this with mice. You take skin off the tail of a mouse, you can reprogram into iPS cells. They're able to make functional sperm and eggs. But to do that last step, you have to make what are called primordial germ cells that are almost finished. But you put them into the testes of a mouse to do the final stages of development, ovaries of a mouse to do the final stages of egg development. You can't really do that with human because you can't get FDA approval to put something that experimental in someone's testes or ovaries to do the last step. That's why people want to make the testicular or ovarian organoids. If you make organoids, maybe you can seed them with the primordial germ cells that you can create. So anyway, if you can make lab-grown sperm or eggs, okay, here's the crazy experiment. So we know in mouse studies you can like crossbreed, you can do all kinds of like really interesting cross strains for research. This would be all done in a dish. Again, I'm not telling anybody you should do this. I'm just pointing out this is possible. You can make so you can make an embryo model. And then from that, get pluripotent stem cells because embryo models also it's interesting. They kind of make stem cells. And it's a little weird, like, what do you call these? Are they embryonic stem cells? But then this isn't really an embryo. There's some classification issues that come up. But, okay, so let me walk you through. You get embryo model that's completely genetically identical to a particular person. You get a male one, you get a female one. You make gametes from the stem cells from that embryo model, and then you put them together. You make another. This is going to be an embryo model. This would be an actual embryo. And then you keep doing that, and you combine the sperm and eggs from all these without ever having it grow up into a person. And you do your cross strains that way of human. So you can't do the experiment where you actually raise babies, wait until like mice, breed them together, but you can do it all in a dish. You can have multiple generations. Now that would be really interesting scientifically because you can kind of do the human studies the way you would do. Well, you could already do that right now with cloning. You can. It wouldn't be your child; it would be your twin. So you would give birth if you're identical twin.
Finally, James, a friend.
Do you really want your Sinead 2.0 to be James's friend?
No,
think about that.
Point. Yeah, excellent.
Yeah, that's fascinating. So you could like simulate thousands of years of millions of years of like human evolution in like just one petri dish.
Yeah,
just running generations.
You're only going from making the germ. And by the way, the primordial germ cells, they appear very early in the embryo models. It's really, and they just pump them out. So it's a fascinating thing of not only creating an embryo model to study development, but it's an unlimited source for primordial germ cells, which then you can hopefully mature in vitro, and then you just start combining them. I need to point out, because a lot of people, as a responsible bioethicist, I should actually be saying, like, well, what's the point of making embryo models? I don't want to just scare everybody and just sensationalize it, because certainly that's possible, but you really have to ground it in, like, what's the point of it? So let me just give you an example of how significant this work could be. So you can make an embryo model that represents a pre-implantation embryo. This is the stage right before you put it in, into the womb for fertility treatment. The vast majority of drugs that women take while they're pregnant have no data on safety for the embryo. So you make embryo models, you can make them all identical from the same cell line. So you have total control over your population of embryo models, and you can put the drugs that people take and examine what's going on. I mean, that's incredible. I mean, that alone is pretty amazing to be able to do that kind of research for just healthier babies in pregnancy, and you're not creating a real embryo. The other thing I should also qualify is all the embryo models I'm talking about don't seem to have full developmental potential. There's always something missing. So I don't mean to say that like they are equal to a fertility clinic embryo. The biologists are very clear. There are certain cell types we're not familiar with. Like it has these other weird cells in it. The spatial organization is not quite right. So none of them will be able to create a baby if you transplant them. That could change in the future with more animal work, and that's an interesting way to think about this. But right now, there's no potential.
So you could significantly reduce the amount of research burden on like fertility clinic, embryo and egg donation. Because I'm thinking if you just took one embryo and then you compared it to 7 and just tested it to be like, okay, is it having the same outcome for this drug? Then you could continue to use those 7 lab-made ones for a bunch of tests.
Yeah.
And you could, that's amazing.
Yeah, and you can generate as many as you want. Now, I worked with a team in Michigan, the state of Michigan in the United States, and I told them when you make, because they can make hundreds of these things, okay? I said, when you make any of them, make sure you always leave out some important component for development. Like don't ever try to make it a complete model. You don't need to for your research question anyway. If you want to study the engine of a car, why would you make a model that also has wheels? Like, just don't complete it. And the reason for that is the state of Michigan has said ever since Dolly the sheep was cloned that it's a criminal offense to do human cloning. And we have to then worry about this as count as human cloning. And it would if it was developmentally competent, because it doesn't matter how you create the clone, whether it's nuclear transfer or this other method, if it could create a baby that's genetically identical to the original donor, that is technically a clone. And in fact, as just mentioned earlier, the embryo models are a truer clone than the nuclear transfer ones because of the mitochondria. In fact, the word clone is a Greek word that means twig, and it comes from horticulture. When you take like a geranium little twig and you can propagate it, that's where the term clone comes from. So technically, a clone is kind of like this embryo model. It would be something that you take from your skin and you can then create another being from it.
Do you think that a lot of the resistance to the concept of cloning is coming from the genuine safety concerns? Like, we don't know what the quality of life of this person will be. Or do you think it's the ick factor?
Oh, okay. So I can answer that. So a lot of my early work was, oddly enough, in this area of cloning, and it came up through sort of like the technique of research cloning for stem cell research. But in any case, let me answer that question in a couple of ways. It's highly dangerous to try to do reproductive cloning for any animal. So you've heard that, they've cloned horses, they've cloned pigs, they've cloned.
I have not heard this. This is news to me.
Yeah.
So sorry.
They've cloned lots of, this is a big business in Texas. You take your prize bulls or your prize animals and you clone them. People have tried to do this with racehorses too, like
I'm
really,
not surprised
really big
capitalism got involved.
prize things. Yeah. And then there's also dog cloning for people who lose a beloved dog. Tom Brady has cloned his dog and Barbara Streisand has cloned her dog. Now, the thing I want to say about animal cloning, pet cloning or anything like that, is what we know from developmental biology, what we know from the people that I work with, is there's no such thing as a healthy clone. The one that's born is the least abnormal. It's the least abnormal, but there's always something wrong. Why? Because the reprogramming process of the egg for the DNA is not perfect. The cell that you put in could have, and most likely will have, mutations. This cell is just, especially older animals, older humans, you're going to have lots of mutation burden. We don't know what you're transferring in there, the quality of that, and you won't really know until, further on. So it's very dangerous. It's like Russian roulette. It's very dangerous for those reasons of the imperfect reprogramming, and then so biologically it's dangerous. Lots of miscarriage, lots of things wrong with the animals that are born. So the ones that make it to adulthood are the least abnormal. Now it's interesting, if you breed a male and female clone, if you breed them together, their sexually produced offspring are fine. That's really interesting. Yeah. So I would say Gen 1 is not great, but number 2 could be okay.
Gen 1
So
taking one for the team.
Yeah, or Gen 0. I'm not sure how you account that, but yeah.
There's something about the sexual reproduction that
So what happens is it resets all the methylation, all the stuff that doesn't get completely reset in the first attempt.
Okay, so it's
so for
highly
humans,
unsafe.
would be very unsafe in terms of you're probably not going to have a healthy baby. You were mentioning the psychological issues.
Ick factor.
Oh, sorry. Yeah. Well, the ick factor, I think people might get over that if it actually did work and you had healthy children, because there was a lot of ick factor around IVF. A lot of people were like, oh, that's so unnatural. They used to call them test tube babies. And as soon as real babies that were healthy were being born, everyone kind of dropped that and they just gravitated toward, I want one.
There should be T-shirts now, like test you.
Yeah.
Capitalism should get in there.
Well, I wouldn't be surprised if they
No, it has in the U.S. Yeah,
Every time
IVF is very
IVF
unregulated.
baby's born, it's like, all right, here's your family T-shirts.
Yeah. So the ick factor, that's also present in human reproductive cloning. But I think a lot of people also worry about identity issues, psychological well-being of the clone. It's interesting because back in the day when people were debating this, someone said, "Oh, clones are all identical. It wouldn't be an individual." And I said, "Are you kidding me? If there were ever a baby that was born through cloning, it would be the most unusual human in all human history. It would be like the most special human being if you knew about that person. So don't tell me that what's wrong about it is you lose individuality. That would be like amazing in all of human history of all the people who've ever lived. Like you are the asexually reproduced human."
Counterpoint: child actors often turn out to be mentally.
Yeah.
So
I'm not tying this to the mental well-being.
Yeah.
I'm tying it to the objection that it's bad for the child because the child doesn't have a unique identity.
Yeah.
Now that unique identity, as I was thinking of it, it could also lead to psychological damage and everything. So it's complicated, but I think that the psychological damage is actually coming from misunderstanding of what the technology can actually achieve. Like people may be expecting you to reproduce not only the genetics of the original donor, but just like their personality and their the talents and what made them unique. You're hoping to Xerox the individual, right? But we know that won't work.
Not even twins are like
Oh,
that.
that's a really good point. Yeah. So identical twins. Yeah. I mean, they were even in the same mitochondria, in the same uterine environment, often treated in similar ways environmentally, and they're still different. It was interesting. There was this whole committee in bioethics looking at the cloning issue during President Clinton's administration, And the person at the head of the whole thing, his name is Harold Shapiro. He used to be the president of Princeton. He was the head of this whole thing about task force about human cloning. And he's an identical twin. He has an identical twin. I said, how did you feel sitting there hearing all these arguments about genetic identity and, how important that is when in your own experience, you know the truth? And he said, it was kind of amusing to hear people's misconceptions about genetic determinism.
It sort of seems like a lot of the issues with brain organoids, cloning, stem cell replication, etc., comes down to some form of genetic essentialism, which is this either presumption, not necessarily an argument, that genetics are exceptional or special just simply because they have DNA. But I think for when you sit there and think about it and you actually ask yourself, why? Why is the DNA the most important thing? Why does that thing make it more special than everything else? I'm not so sure that the answers are so clear.
Yeah, there's a version of that that's called genetic determinism. So like everything about your genes determines everything that you'll be and it'll totally accomplish,
false.
right? Yeah, so, and I think here's a very simple thought experiment for the everyday person to do. Okay, so imagine have in mind someone that you really love, that you're in a relationship with, and that person unfortunately passes away. Imagine a doctor coming up to you and saying, I don't know why you're so sad. I don't know why you're grieving. We can clone that person and bring that person back. Would you think that you are actually going to be able to bring that person back? There's no way, right? Because what made that person special to you were the things you went through together, all the experiences they went through. That wasn't just like their genetics. So that's why it's just so, I think, off target to talk about, the risk, the moral objections to human cloning is something about, reproducing identity. Because just do that simple thought experiment. You're not getting that person back. There was a really beautiful essay written by the former president of the Hastings Center for Bioethics, Tom Murray. And very unfortunately, during the whole cloning debate, his daughter in college was murdered. He wrote this really moving essay. It was called Even If Cloning Worked, It Wouldn't Bring Her Back. So that's where I kind of got to say, yes, look, realistically, this is think it through. Don't be worried about that issue. Be more worried about safety. Be more worried about more tractable issues that are probably realistic and not this fantasy about, reproducing people and everything about them.
So for now, we don't need to worry about cloning people and cloning babies and making an army of giant clones and brains in vats. But what is exciting, I think one area that we haven't quite touched on, but I do think is super exciting, is the way that these brain organoids can really contribute to the gap in pediatric research for child developmental disorders, because there's such a small pool of children with rare genetic conditions, and it's so difficult to subject them to so much research that, we have a massive gap. And but now you can get people who have had those genetic conditions as adults, and they can consent and provide full and valid consent to have basically their whole childhood just regrown in a jar. And I'm really excited about that, the therapeutic potential of this, not necessarily the reproductive potential.
Yeah, that's a really good point. I wish that more often people thought about the opportunities to actually not have to use pediatric samples and kind of, the example you gave is why can't you just wait till they're old enough to give consent? So a couple of things. One is people just maybe it's a condition for which all throughout their lifespan, they lack decision-making capacity. 'Cause that's true for a lot of neurological conditions. So unfortunately, you kind of never get to the point where the person can give consent.
Or if the child passes away during childhood.
Exactly. Yeah. So often for neurological disorders for children, genetic base, usually for the majority of them, they're fatal. Yeah, that's quite unfortunate. So that's why there's some desperation about finding out what's going on. Use all the tools in your toolkit. There's X factor, yes, but as I said earlier, you have to balance it against what is the benefit. You can't just look at the bad. Everything has a good and a bad. And you can't just look at the bad. Even if you're making weekend plans, you got pros and cons. Who would only look at the cons? You got to also look at the pros. So everything's got pros and cons.
So don't worry about brains and vats, but worry about doctors stealing your pee.
And maybe James will finally have a friend.
Oh, well, let's not get too optimistic about that. So that comes from the person who has admitted to going for karaoke with me. I'm going to put that.
Many times.
Yes.
But I'm look, mate, I'm there for the vocals, not necessarily the brain. Maybe we can clone
the vocal cords. You get a vocal cord organoid.
I'm just imagining like this little brain that's like singing in the back and it's like doing little dance moves.
That brain's living its best life.
You've got like a bowl of boba and it's just like driving to the music, like with the vibrations.
You can take it around. You can actually preserve it in like a little keychain, so you can carry around your friend. Hello,
Imagine
Tamagotchi.
having a little organoid in a
You can
in a keychain.
literally wear your friends.
Yeah.
Jewelry.
Okay, I think that is all the time we have for today. This has been such a fascinating pod. I feel like I've learnt a lot. A little bit grossed out, also pretty excited.
Been so interesting.
Let us know where we can get James a friend, and
Right,
we'll
right.
see you around.
Well, welcome to my world. Thanks. It was fun.
Thanks for listening to our new podcast. Subscribe online and tune in every first of the month. In the meantime, listen to CBME and you and be savvy.